Thank you. Thanks for the opportunity to present to you, although I'm a bit shaken by that threat to coffee. I'm not quite sure. I thought my biggest problem today was having to wear a tie, but the coffee is terrible. So I'm going to talk to you about our platform, which is a synthetic biology platform that allows us to create totally unique, patentable synthetic promoters and provide some great tools to actually start to control gene expression in some very special ways. And whilst the whole industry for the last several decades has been focused on using viral promoters to control genes, we've come up with a capability to custom design promoters that are very precise in terms of their mode of activity, in their site of activity, and start to do novel things in terms of inducibility, etc. And really start to focus on some of the next generation gene therapy and cell therapy applications that can be enabled through improved gene control. So we're a team now of 35 people based in Edinburgh. We're growing rapidly. And our model is we're not developing our own therapeutic products at this stage, but we're developing our technology, patenting it and licensing it out to other mainly gene therapy companies. And that's shown by this slide here, which reflects our latest deals that we've done. So we've now got over 16 separate commercial entities that we have deals with, and over 26 commercial programs that we're working on. So in the licensing model, that then equates to over $350 million worth of deal value. The blanks are those companies that won't let us use our names, so some are undisclosed. But our vision is to be able to say, well, if you can control genes, if you can create synthetic promoters to control genes in a constitutive basis to make them precisely active in target cell type, to control them for size, to control them for strength of activity. That's great for monogenetic diseases, but then there's more complex diseases that we're developing platforms that allow us to induce and turn on and off gene function. Being able to turn genes on and off is great, but the next question is, what about more complex applications like multi-gene cassettes, gene pathways, and logic gates? We've created concepts that will allow us to create multi-gene cassettes and allow us to create logic gates. And then the blue sky thinking of where this technology leads is really the fourth step here, which is then genetic circuitry and pathways and being able to engineer complex and deliver complex genetic circuits for complex diseases. Just some quick data just to show how the performance of the technology. This shows performance in liver cells on the far left. Then you can see our synthetic promoters in an in vitro liver cell line, showing that the black are our synthetic promoters, the red is the control promoter. This control promoter shown here is actually currently in a phase 3 clinical trial. And so we've shown that we can beat it in vitro by about 75-fold greater activity. That's then been translated by our partner uniQure into in vivo data, showing that we get up to 50-fold higher activity from our promoter than something that's currently at use in the clinic. We're now starting to work to build multi-selective promoters. So the question we've been asked is, well, how can you start to create some promoters which are active in a series of cell types of choice and exhibit differential expression in those target cell types? So whether that's different muscle cell types or whether it's liver versus cardiac cells. And so we're now building multi-promoter parts that will allow us to set up multi-modular promoter activity. And this is another quick slide which basically shows the expression range that we typically see with our promoters. And we're using this to create multi-gene cassettes where the stoichiometry of protein expression is important to functionality. So you can select a unique promoter to differentially express genes to different levels for particular purposes. I mentioned the inducible promoter platform, so in a bioprocessing environment, then we're regulating toxic genes for things like AAV and lentiviral production, and we've been able to show that when our inducible promoters are uninduced, they're totally silent, and we can control a toxic gene without impacting cell viability, and when it's on, then it's strongly on. And we've now initiated a program to start to take some of these concepts and tools and apply them to a more therapeutic setting, and we'll be announcing more of that over the next 6 months. And then finally, what we're also doing is now collaborating with some early stage research institutions to start to enable the next generation of therapeutic products. And there's 2 examples. This is one which is in neuronal cell populations, where we're working with a team out of UCL to develop a therapeutic for juvenile Parkinson's disease. This is interesting to us because having the gene expressed in the appropriate cell type ameliorates the disease. Having the gene expressed in an off-target CNS cell type actually kills the animal model. So tight regulation is required here. And we've got another program where we're doing something similar with hematopoietic cell lines to create promoters which are differentially expressed in a range of hematopoietic lineages. So that's a quick summary of the technology and what we're about.
Great, thanks, David. I won't ask you the question I've asked every other company today, which is whether gene therapy's arrived. The answer's been yes. So why don't we talk then instead about what the unmet medical need that's remaining and why your promoter technology, your foundational capabilities actually address that need.
Yeah, so I think, yes, I would agree. I wouldn't disagree from what everyone else has given you an answer that yes, it has arrived. However, I'd say that great advances have been made, huge advances in capsids and addressing safety concerns and in developing the industry. But I think we're starting still to have questions over, well, is this a treatment for life? And if this is dosed into somebody and we see unintended effects at some point down the line, then could we have the option to turn a therapy off? And also there's some diseases where you wouldn't actually want the gene to be expressed continuously. And so being able to have transitory expression of the gene or having an option of when it's turned on or turned off, I think can be enabled by some of the concepts that we're developing. I think also if you look at clinical trials, many of the questions are asked is, well, if you look at the same therapeutic setting, essentially the same capsid, the same gene, but you're seeing quite differences in patient response, both within a trial but also between trials. And what's at the root cause of that? Where is that difference coming from? And how do we address that? And that's why I think there's a lot of scope to improve gene expression, improve the specificity of gene expression, the amount of gene that's being expressed, to bring everybody over that threshold. But also if you can create a promoter where you can dial up or dial down expression, you can make sure that for a particular gene, for each patient, they are within a therapeutic window.
And what would be an example, a practical example of, let's say, a blue skies outcome where your technology works very well? Where might you want to turn things on and off?
Well, I'll give you one example, certainly for therapeutic threshold that we've been approached about and have discussions around, and that's something like Rett syndrome, where having too much of the protein expressed is as bad as having too little. And obviously trying to use transfection efficiency or using the capsid or to actually achieve the right therapeutic window for each individual patient is quite a significant challenge. So this is why we're really interested in developing therapeutically applicable inducible promoters so that we could basically dose a patient at a high genome equivalence, but then dial up the amount of activity of that promoter of that gene based on the inducer and the concentration of the inducer. So the fact that we've been able to create inducible promoters which are dose-dependent, dose-responsive to the inducer is very important there. Obviously, it's now building what we've shown in a biomanufacturing setting into something that's appropriate to an in vivo setting.
Yeah, I found interesting your slide with all your partners. There are many of the companies who've been talking about promoter science, and now I find out that it's Synpromics that's under the hood. So I'm curious on how you got so many partners. What is it that you're doing differently from other companies, if there are that many, who are focused on promoter science as well?
Yeah, it's a good question. I mean, people have been engineering promoters for decades. Since the birth of the industry or biotechnology, people have been engineering promoters. But I think where we clearly differentiate ourselves, and the flash of inspiration that our scientific founder had, was to say, well, with the genomic databases that are now available, you can actually analyze and understand transcription control elements on a massive scale. And so, for example, for a liver promoter, we've developed our prompt system, a bioinformatics database, which is now terabytes in size, and has machine learning algorithms built into it to mine that database for elements that will control a gene on a very cell type specific basis, and take all of those elements, and that enables us then to design totally unique sequences from combinations of those elements, screen those both in vitro and in vivo to understand the activity, understand the relationship, understand what works and what doesn't work, repopulate that information back into our bioinformatics database, and use our algorithms to search it again. So that at the end of a series of those cycles, you've come up with a sequence that nobody would, if they were sat in a lab looking at sequence, that nobody would have thought of putting those elements together or of building it. And so we've done it on an industrialized scale. And at the end of the day, we're able to create something that is custom designed to the specific design brief that's required for the specifics of the therapeutic, or the specific of the disease, or the specific of the patients that are going to receive that disease, or the specifics of the delivery system that's going to deliver the gene. So we can code in all of those variables and get the optimal at the end of the day. And the scale with which we can do it is so vast that whilst there's always a place, and people will always love to tinker and play and come up with their favorite sequence that they've adjusted in various ways, I think the power of the big data and our ability to analyze it and code it is unique.
Right, and in focusing on that elegant slide with all the partners, could you talk a bit more about how you select your partners first, and second, that difficult question of whether you do it on your own with your technology, or you partner it out. So we've seen big companies like Alnylam wrestle with that dilemma. How do you go about that?
Yeah, so maybe it's a personal thing. I love collaboration. I don't think you've got the fount of all knowledge in any individual or any small group of individuals. And so if you've got a transformative technology, I almost feel that there's an obligation on us, particularly in genetic disease and in some of the therapies that are crying out for treatments that are currently unmet. The more that we can do to enable the industry, the better. We've seen what the capsid guys have done to go back into research and academic institutes, solve problems, come out with something new, and then help to drive the industry forward. If they'd kept all of that tight to themselves, then we would have not seen what we've seen today, and we would not have advanced as much. And based on the power of the technology, there's loads more that our technology can enable and do than I could ever do myself, or our team could ever do, or we could ever raise the finance to do. So there's something about me that loves the collaboration, loves interacting with people, loves enabling other people, and loves to see success and patients treated. And so we will continue to do and drive that. However, I'm also aware we're running a commercial business. We're trying to create value for our shareholders. And I know that as we start to generate more and more data and get more and more compelling evidence for what we're able to do, there will be kernels of opportunity that arise from that I'll think, well, actually, that looks so interesting, such a great opportunity, I'm going to keep that one for myself. And we will raise the money that's necessary, and we'll build the team that's necessary and start to do that ourselves. So whilst as a bootstrapped UK-based company that doesn't have access to the same capital as you all have over here in the US, we've had to be lean, we've had to be efficient, we've had to maximize what we've got, and that's served us well. But we are continuously looking for that opportunity to take some of our greatest novelty, bring partners on board, raise the investment required, and take some of those products through to the clinic ourselves. And I mean, the things we're doing with UCL, and we're in discussions with a lot of other academic centers, because the innovation is still coming out of the universities, coming out of the academic clinician groups, and we're starting to work more and more with these groups, because we want to enable not just the second generation gene therapy, but the third generation gene therapy. And by coming in alongside these teams of people and seeding our technology into the labs, we think we can enable that and take products in ourselves that we can then take forward.
So just with my final question, I want to follow up on that in terms of innovation in the future. You're obviously levered to the future of gene therapy. So what do you see as major innovations or next innovations, and how will your technology help solve or help solve the problem and help with the improvements?
So I think, I mean, we're doing well interrogating and looking at monogenetic diseases and coming up with therapies in those areas. But we know that everything we learn tells us that the genetic basis of certain diseases is becoming ever more complex. And so we're going to have to need ever more complex solutions to some of the larger diseases, some of the lifestyle-induced diseases. And I see our technology as being an enabler of that future generation of more complex genetic circuitry that's going to be required to be built.
Thank you, David.